Good afternoon and welcome everyone. My name is Delphine Costa. I'm the CEO of the Life Science Cluster, and I'm very happy to welcome you this afternoon for this webinar. So just a few words first about the Life Science Cluster. So the Life Science Cluster, we are a national cluster with more than 120 members, spanning from pharma, biotech, diagnostic, and bioresources. And our mission, it is to connect companies, academia, and public sector and public stakeholder to accelerate innovation and strengthen Norway's role within life science. And that's exactly why we are here today, to explore how AI is reshaping drug development. Drug discovery is, and has always been, quite costly, time-consuming, and risky, and AI is now offering huge opportunity to change that by making the process faster, smarter, and ultimately helping bring medicine to patients faster. I'm very happy to introduce our speaker today, Petrina Kamya. Dr. Petrina Kamya, you're the VP Global Head of AI Platform at Insilico Medicine, and you're also the President of Insilico in Canada. You bring extensive expertise at the intersection of medicine, data, and technology, and you will share your perspective on how AI is already transforming drug development. So just before you start, I just want to all let you know that we will open for a Q&A session at the end of this session. So you can either raise your hand and we will unmute you, or you can write your question within the chats and we will read your question. With that, I'm very happy to welcome you, Petrina. The stage is yours.
Thank you so much, Delphine. It's an honor and a pleasure to be speaking to you all today on behalf of Insilico Medicine as a pre-webinar to the talk that we'll be giving at the In Life Sciences Cluster conference in November in Oslo. So if you have any questions that you have been burning to ask during the webinar but not able to, if you're going to be there in Oslo, I'd be happy to meet you face-to-face and answer those questions in person. So I'll just begin by sharing my presentation with you all. There we go. I hope you can see my slides.
Yes.
All right, so I've been working in this space for quite a while now, and when I started about 5-6 years ago, a question that I was always asked, or I often heard being asked, and I still hear this today, is how the application of AI in drug discovery has brought value to patients. And I'm focusing on drug discovery because I'm talking about the early stages of drug discovery and development. Yeah, okay, the next slide. So, and when I'm talking about the early stages of drug discovery and development, I'm focusing more on which of the first couple of stages actually brings value to towards bringing drugs to patients. And another question I often get asked is what's next in terms of the application of AI in drug discovery and development. Now, for those who are not familiar with Insilico Medicine, we are a AI drug discovery company. We started off by building the technology before we started to build our pipeline. The technology itself came out of Johns Hopkins University in Baltimore, Maryland. And as far back as 11 years ago, we started to take deep learning algorithms that had worked in other industries and try to adapt them towards some of the challenges associated with drug discovery and development, starting with target ID, going all the way up to bringing medicines to the clinic. And to that end, over the last 10 years, we've built an integrated AI drug discovery platform that we call Pharma.ai that has capabilities in target discovery, small molecule, peptide, nanobody, antibody, design and optimization. And we've started to work on improving clinical trial optimization as well. So as we've been establishing this platform, it was very important for us to demonstrate that the platform actually works. So not only did we license out the tools, so we have a number of clients out in industry, but we also started to build about 5 years ago our own internal pipeline of therapeutics. Today, that consists of 31 internal programs against 29 targets in various different disease areas. Our lead program recently completed phase 2A successfully, reaching both the primary and secondary endpoints for a disease that has no cure and has high unmet need. This is idiopathic pulmonary fibrosis. We have 5 other programs in phase 1 and 10 programs in IND-enabling studies. And we were able to do this all in under 5 years, so just about 5 years. As we've been around for, well, relatively speaking, a long time in this space, we've published a lot of our findings as well. So we have over 200 peer-reviewed papers that we've published, and we were one of the first to publish on the use of generative AI in this space. So very strong foundation in this space, and we understand that in order for you to appreciate the value that this can bring, we also leverage our tools, and we have our internal pipeline. So that's the company in a nutshell. And this is a snapshot of the technology that we've built out over the last 11 years. And what I want to show you is that with these tools, I'll describe a few of them to you, what we have been able to achieve in the last 5 years in a big way. So I have 3 stories to tell you in the form of our internal pipeline, and then in also small ways. And these are with collaborations that we've had with academic labs in the space of drug target identification and drug repurposing. So the first tool I'm going to talk to you about is PandaOmics, which is a platform that we have developed and built to identify targets, biomarkers, and indications. It plays a crucial role in our program strategy from the start, helping with our, from the start, as in identifying the targets, understanding the pathways that are perturbed in a disease, all the way up to our biomarker strategy for clinical trials. And what makes this platform unique is that it leverages multimodal data, something that as a human being is very challenging and difficult to do. This includes omics data, where we look at multi-omics data, genetic data, we look at RNA-seq, single cell data, metabolomics, methylomics data as well. As text data, so we look at publications, we look at drugs that exist, that are going through the clinic as well, clinical stages, so that we take this data-rich, driven approach towards decision-making for our internal programs. Whether or not that's a novel target or an existing target, we always leverage this platform to help us make decisions regarding which targets we're going to pursue in our internal programs. And our clients do this as well. Once we've identified a target, the next thing is to design a molecule that modulates the behavior of that target. And one platform that we use to do that is Chemistry 42. This is a platform that is a multi-agent reinforcement learning tool for small molecule design and optimization. It leverages generative AI for ideation and for and a comprehensive reward function for multi-parametric optimizations. Again, we use Gen AI for ideation, and then we have that reward function that's very, very customizable for that multi-parametric optimization. What this makes this platform unique is that it dramatically reduces your DMTA cycles by helping you or us design molecules with the types of properties that we want the molecules to eventually have. So we typically only synthesize in the order of hundreds of molecules as opposed to thousands of molecules by the time we get to the development candidate phase. Okay, so in the same breath, we have a platform that's similar, but for the design and optimization of biologics. And this platform leverages graph-based AI, large language models, and diffusion models on the ideation part. And then we have property optimization and computational biology approaches to improve on the properties of the biologics that we design. So again, this is peptides, nanobodies, and antibodies. So very much the same workflow for small molecules, but now this is adapted for biologics. And of course, where would we be without data? We have, I guess we did this in 2020. So one of the first of, I would not say fully automated, but automated labs that is primarily focused on target validation and then the hit to lead workflows that we now call LifeStar 2. This features 6 automated modules that work in concert to generate data that informs on our target identification. And then once we have identified a hit and synthesized it, our ADMET predictions that we make on the molecules that we design and optimize using our platforms. So this lab actually helps us shorten that DMTA cycle quite a lot by allowing us to leverage the data that we generate for each of our programs to improve on the predictive properties of our algorithms as the programs are progressing, as well as help our target identification tool improve on its target ID capabilities by having that data, validation data fed back into our algorithms as well. So what have these platforms helped us do? And this is the complicated story, or the more involved story. Because we use all of our platforms as well as our lab, what we have been able to do is significantly shorten the time it takes to go from discovery all the way up to nominating a preclinical candidate quite dramatically. Whereas this process typically takes, and I'll get my laser pointer out here, 2.5 to 4 years. The longest time it has taken us to date is 18 months, and the shortest time is 9 months. And we've done this not 3 times, but over 22 times. So we have 22 development candidates in our pipeline, and 10 of these programs have received IND clearance, et cetera, et cetera. So we've demonstrated by leveraging that platform, Pharma.ai, we've been able to significantly reduce the time it takes to get these drugs into patients. And of course, we've published all this work. This is a bit of a busy slide. It summarizes basically all of the work we did for our lead program, from the moment we identify the target, all the way up to completing phase 2A trials. A busy slide, but we've also published 2 Nature papers. So if you would love to learn more, and we go into great detail regarding how we were able to do this in those 2 Nature medicine papers, from target identification to phase 1, and then from phase 1 to phase 2. All the data is there, all the workflows, everything that we did, we have shared with you. One thing I want to also highlight is, in addition to identifying, that target and implicating it in fibrosis, TNIC and fibrosis, we were also able to leverage our Pandomics platform to identify a host of other indications that the target could be leveraged in to de-risk our lead program. They're all fibrotic programs, so it's a pan-fibrotic, the molecule that we've designed and optimized. And we've also been able to validate using our automation lab, that this particular molecule is also implicated in a lot of hallmarks of aging, so pathways associated with aging. All right. And this slide is just a snapshot of some of our more advanced programs. You can see that we've also out-licensed some of our molecules. I believe we're one of the only companies that has managed to do that. So we've out-licensed 3 of our internal programs to external partners. But you can see that there's a wide range of different types of therapeutic areas. So we're in obesity and metabolic diseases, we're in pain, and then we also have oncology and fibrosis. We have 1 anti-infective as well. So short answer to the question, a quick answer to the question, does the adoption of AI in the early stages of drug discovery really bring value to patients? And the answer that I would give is yes. The speed with which the application of AI is bringing drugs to the clinic is quite amazing. But that's not everything, right? Yes, the molecules also need to be safe and efficacious. And that's something that AI can help you do in a very short period of time. All right, so how else is AI helping get drugs to patients? So for this, I'll tell 2 short stories. One involves ALS and our collaboration with labs out of Johns Hopkins University and Harvard University and a startup based in Taiwan or China. So for those of you who are not familiar with ALS, it stands for amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. It's a progressive neurological disease that affects nerve cells in the brain and the spinal cord. What happens over time, and not a very long period of time, is that you lose control of your muscles, starting with the limbs, spreading to other parts of the body. And as the disease progresses, you just slowly lose control of all of your muscles, ultimately leading to suffocation and death. I'm sorry, these stats are based on North America, but please bear with me, as this is not unique to North America. But it affects about 1 to 9 in 100,000 people in the US, and it costs about $1 to $1.4 billion a year in the US alone, and there are no known cures. So in collaboration, like I'd mentioned before, with Johns Hopkins University, those Harvard, the Mayo Clinic, and Answer ALS, they came to us as academic labs that don't have very big budgets and asked us if we could leverage our tool to help them identify targets that they had not seen before implicated in ALS. And then of those targets, can we identify a molecule that they can repurpose quickly and take to patients? So go straight to a phase 2 clinical trial and see whether or not it could have any positive impact on the patient's quality of life and even help reverse the progression of the disease. So the study exemplifies the full potential of leveraging a multimodal tool like Pandomics for target identification, that was paired with in vitro validation that we didn't do on our own. It was done by domain expertise, people who had these assays and the domain expertise in this disease area. So this is just a breakdown of what we did. We took publicly available data sets and we did it, curated them. We took also the data from the Anti-ALS project and used all this data to identify targets in Pandomics. And then we filtered those targets based on their criteria. So they had to be targets that were not necessarily novel in other disease areas. So we didn't have to go through the whole preclinical testing, animal and phase 1. And they also had known drugs. So it was easy just to purchase the drug and retest it in phase 2 clinical trials. And all of the hypotheses that we came up with, they tested in their Drosophila eye model for ALS. And what we were able to do, this was in a period of months, was identify 28 potential therapeutic targets that satisfied their criteria. Of those 28, 8 were verified as unreported genes. So some were more or less known, but 8 were completely novel genes where their perturbations strongly rescued eye neurodegeneration in their Drosophila eye model. And based on that, they started a phase 2 clinical trial. This was about a year and a half ago. We're expecting the readout any day now on patients that have ALS. So in 18 months, it's a relatively straightforward and simple use case, but we were able to help and get drugs that already been vetted for, tested in patients who are suffering from the disease. The next story I'd like to tell you is similar in terms of the end goal, but involves a completely different disease. And this is endometriosis, which is a chronic inflammatory condition where endometrial tissue grows outside of the uterus. Nobody knows to date what causes it, whether it's genetics, environment, immune dysfunction, estrogen perturbation. But what is known is that it causes a significant burden on women all around the world. Here I have a stat about women from all over the world, not just North America. It astonishingly affects 1 in 10 women worldwide. And the economic burden, in the US alone is $80 billion. And there's no causal treatment options available. So we partnered again with academic labs with domain expertise in this disease area. And what they wanted us to do was to leverage publicly available data to try and identify targets that could be implicated in endometriosis that are not involved with hormonal pathways. They're done with perturbing the hormonal pathways in women. They wanted something that was not hormonal, mostly associated with immune dysfunctions was preferred. And then once we've identified those targets, are there any existing drugs that they can repurpose and then test quite quickly to see the effects, whether or not the effects are true. And they had, again, the in vitro and in vivo models to be able to test any hypotheses we came up with. And once again, in a very short period of time, we were able to identify 2 targets that were significantly upregulated in the data sets that we used to identify the targets using Pandomics in patients suffering from endometriosis. what was key here is that the pathways that these two targets are implicated in are not related to hormonal pathways. They are related to the immune system. And neither of these targets had been implicated in endometriosis. I'm missing a fourth piece here where we actually identified a drug as well that could be repurposed, but I shortened everything just for the sake of time, and just left it to the evidence supporting the implication of these two targets in endometriosis. So we came up with the theories, they went out and tested them. And in vitro assays where they used anti-GBP2 and HCK antibodies and compared the staining in tissues from women who suffered from endometriosis, so both ovarian and peritoneal tissue samples, compared to healthy women, they saw that these genes were overexpressed in the tissue samples that came from women suffering from endometriosis. So then they looked at siRNA knockdown of these two genes in mice and found that when they knocked down these two genes, there was a decrease in the lesion volume found in the mice compared to control, and a decrease in the lesion weight. And they also looked at the increase in apoptosis. So when they knocked down these genes, they saw an increase in apoptosis of these lesion cells, the cells of patients that had endometriosis compared to control as well. These are simple stories, but it shows that there is an impact that can be made by leveraging AI in drug discovery and development that doesn't have to be super expensive or super fancy. It could be just as simple as using known existing data and trying to find patterns that have previously been missed before or not been seen before. So basically, speed, innovation, and reduced costs. These are the three ways that AI is helping get drugs to patients today, and leveraging AI in the drug discovery phases of drug discovery and development. So we're able to significantly reduce the time it takes to get drugs to the clinic. By leveraging AI, we're able to uncover previously unknown connections between genes and diseases, identify drugs that can be repurposed. And by making and testing fewer molecules and leveraging the data that we already have out there, we can provide significant savings to companies that are working in this space that should then be passed on to patients. And I wrote that R and put should because I'm not going to tell companies what to do when it comes to pricing. So what's next? I'm sure a lot of you have heard about agentic AI. And I'm sure a lot of you may have heard of autonomous lab in the loop, moving the humans out of the whole design, make, test cycle for drug discovery and development. I was recently asked to speak about this, the use of agentic AI and what I thought, and share my thoughts regarding what I thought about it. And what's the status and the promise of agentic AI, and is it really where we're going in the future? Just for everyone's sake, I will share with you what my definition of agentic AI is. And hopefully this will set the stage for how I believe everyone should think about, or most people can have your own opinions, should think about agentic AI in our space. So agentic AI is basically a system that is designed to pursue its own goals autonomously. It can perceive, plan, take actions, learn from feedback, and adapt. Now, I would like you to bear this in mind when you think about the typical stages of early drug discovery up to the point of nominating a preclinical or a development candidate. I think for the latter stages of drug discovery and development, in terms of optimizing clinical trial protocol, ensuring that if a patient is not adhering to the protocol, make a decision around what to do about it, right? So we inform the doctors, or we inform the patient and tell them that this is important, that you have to do blah, blah, blah. That kind of autonomy, I think, is kind of obvious, but so I'd like you to focus on the early stages of drug discovery and development. And I've broken these down into four main pieces. So disease hypothesis, target, identifying hits, hit to lead optimization, and then lead optimization. And I'd like you to bear in mind the fact that agentic AI is a system designed to pursue goals by itself, perceive, plan, take actions, learn from feedback, and adapt. How comfortable would you be knowing that an agent is responsible for all of those initial stages of drug discovery and development. Okay, I'm going to leave it at that. Happy to answer questions, happy to talk about this either today or when if we meet in Oslo. But this is something that apparently is coming up next in our space. So to conclude, I hope I have conveyed to you at least a few examples of how leveraging AI in the early stages of drug discovery and development can bring value to patients further down the line in the value chain for drug discovery and development. And there are clinical stage drugs that are actually being taken by patients that are showing efficacy and the safety profile that we want drugs to have. Yes, so yeah, there are drugs that are getting closer to patients that have been designed by AI. And with regards to what's next, I'm not sure. But there's definitely a lot of buzz and talk out there around autonomous agentic AI and the role that it's going to play in our space. And like I said, I'm happy to hear your thoughts and talk to you all about that. And with that, I'll say thank you for your time, and I look forward to having a discussion with you.